Welcome everyone. Let me introduce my PhD topic, which is antimicrobial therapeutic drug monitoring in critically ill patients by clinical pharmacists. My name is Eszter Gulyás. I'm a pharmacist and working in the University Pharmacy Department for 6 years. And during the recent years, I became interested in critically ill patients' clinical pharmacy, and my vision is to facilitate strong cooperation between the clinicians, the laboratory medicine specialists, and the clinical pharmacists, and to promote TDM-guided antibiotic therapy in critically ill, and to become an expert in the utilization of these results. I have two ongoing projects, and the goals are the following: to investigate the effect of beta-lactam TDM in critically ill patients, and the second is to evaluate the pharmacokinetics-based guidance of colistin therapy. The title of my first project is Guiding the Administration of Beta-lactam Antibiotics by Therapeutic Drug Monitoring Improves the Probability of Attaining Clinical Cure in the Critically Ill Patients. The methodology that was used is systematic review and meta-analysis. The background of, this is the current status of my manuscript. The manuscript is under edition, and we hopefully will submit in a month. Our target journal is Critical Care Medicine, and we already sent out the pre-submission inquiry, and I will gladly share you during the discussion part of their reply. The background of the first topic is that around 70% of critically ill patients receive antibiotics while they are admitted to the intensive care ward, and beta-lactams are the most frequently prescribed. That's why we targeted this group of antibiotics. And this patient population is completely different to other not severely ill or healthy volunteer patient population, and we are uncertain about the pharmacokinetics of these drugs in these patients, and that's why therapeutic drug monitoring came as a specific tool to individualize the therapy. This means that this is the measurement of drug concentrations in blood in the specific intervals and the adjustment of therapy upon these results. But we don't know its effect and ended value of the therapy and the patient outcome. That's why we would like to evaluate the impact of TDM. Our clinical question was, is TDM-guided beta-lactam antibiotic therapy more effective than standard of care beta-lactam therapy? So we compared TDM-guided adjustment of beta-lactam therapy to standard of care therapy. And we had 4 outcomes, and clinical cure was our primary outcome. We performed a systematic literature search, and around 3,000 articles were found, and 7 found to be eligible for data extraction. Here are our results. The primary outcome was clinical cure. It seems to be the most promising result because, as you can see, it favors TDM in connection to clinical cure 2 times higher in the TDM group than compared to standard of care group. It is both statistically and clinically significant result. Our second outcome was microbiological eradication. And as you can see, we performed a common effect model and a random effect model to compare whether it would be significant or not. We can see that there is a trend because all the diamonds are on the favors TDM on the Fowler's plot, and you can see that the odds are almost nearly 2 times higher to favor microbiological cure in the TDM group. And here I would like to present our other outcomes, which was hospital mortality and ICU mortality and length of ICU stay. You can see that we cannot declare either it is favors any of the groups. but we can assume that mortality is more complex and complicated outcome than to be influenced only by TDM therapy. As for conclusion, we can say that using TDM is beneficial for increasing clinical and microbiological cure. Well-designed randomized controlled trials are needed, and standardization of protocols and clinical endpoints would be essential. For clinical implications, I would like to say that TDM-guided administration of beta-lactams is associated with improved clinical and microbiological cure. And for research implication, as I said, RCTs are needed with greater sample size and standardization of clinical protocols for PK/PD index calculation. And what would be the best if we could count cost effectiveness of this investigation. The strength of our study was that this was the first meta-analysis done in this topic with beta-lactams. We used quantitative outcomes and hard clinical endpoints, and these endpoints bear direct relevance to clinical decision-making. The limitations were the small sample size, both observation and RCTs, and the heterogeneous population. I would like to move on to my second project, what is clinical evaluation of the pharmacokinetics-based colistin therapy. This is a protocol writing in this phase, and the background of this topic is that colistin was used decades ago, and then it was abandoned because of its narrow therapeutic index and nephrotoxicity. But in the last decades, due to the emergence of gram-negative superbugs, especially Pseudomonas aeruginosa, it was taken back in routine daily clinical therapy. But we are lacking the therapeutic drug effect and the pharmacokinetic of colistin, and no methodology has been available for TDM of colistin. So we would like to evaluate the pharmacokinetics-based guidance of this therapy. A little bit about the pharmacokinetics of colistin. It's a very complex drug. It belongs to the group of polymyxins, and it has an inactive product, which is colistin methanesulfonate, which transfers to colistin in the body. It has a wide distribution. It has a 50% protein binding and 2 to 3 hours half-life. It eliminates with glomerular filtration. You can see here our schedule of our project. We have developed the analytical method in the laboratory medicine department. The technical validation of the method has been done. The stability of the studies is under investigation, and we are obtaining now clinical samples, and we have ethical approval, of course. And we created a antibiotic therapy form to be easily follow the administration of this drug, and this is enclosed to patients' documentation as well. You can see here the colistin molecules and internet standard ion chromatograms as a proof of the methodology existing. And our clinical question will be how should colistin be dosed and monitored to improve its therapeutic efficacy in critical care. And we will compare the administration of colistin by setting the individual pharmacokinetics targets and compare the standard of care therapy. And the clinical implication of this will be optimization of colistin therapy. And I would like to emphasize the multidisciplinarity of this work and all therapeutic drug monitoring because in Semmelweis University, the laboratory medicine specialist, the clinical pharmacist, and intensive care clinician working together like this therapeutic drug monitoring cycle to improve patients' care. And I would like to finalize my speech with that with these two ongoing projects, we were able to submit 3 abstracts for 3 conferences, 1 international and 2 national We would like to share our results. And thank you for your attention with Marie Curie's quote.
Thank you, Eszter. Well, you mentioned cost effectiveness, and although you said that you were not able to do any analysis, but I'm sure you've done some readings in this topic, and I'm just curious that what is the cost effectiveness of therapeutic drug monitoring? It's not cheap, is it?
Well, thank you for the question. It's not cheap, and it's very hard to measure. As a hospital pharmacist, I'm taking care of drug supplying, and I know how it's hard to follow with our existing informatical background to follow exactly the cost of one drug or one treatment or one therapy. Although, on the other hand, I can see that not in the intensive care, but we have individualized dosing system. We have an automatical drug dispensing system which helps us to calculate cost effectiveness. But we cannot apply for this in critical care because the dosing and the administration of drug is completely different to other wards. So really, I cannot give you an exact answer how they measure because I haven't learned about that. But yeah, that would be something which we should investigate on internet evaluate.
Yes. But in your second study, so what is the way of patient recruitment? How do you get the patients? And is there a diversity in patients? Is this easy or difficult?
It is difficult. I am a member of a bigger working group who is working with chronic critically ill patients who are on the ward more than 7 days. So this is our basic recruiting protocol. But this study is concentrating only on colistin right now. And in the recent months, only we have had 3 patients who received colistin. It's quite weird because like 1 year ago, tens of patients received the antibiotics, but you know, we cannot calculate it before in advance. So this is our recruitment policy.
Human trials, registries, things, I mean, that's not easy. I mean, as many of the people in this room already appreciate that. So when do you project to submit your first paper? What do you think?
I think in the middle of June.
Okay,
yeah.
Go ahead.
I have one question about that. First of all, the beta-lactam, it's a huge group.
Yes.
What is the most important point in the beta-lactam systems?
And the most important is that, for me, that we should reach at minimum 4 times the minimum inhibitory concentration during dosing administration interval.
Does it mean you have got together the clinical data, the clinical microbiological data, and the level of the drug? Yes.
Yes, and it's also important to obtain an exact MIC.